Distinguished professors, dear colleagues, in my presentation I will be talking about epigenetic mechanisms, and then I will also talk about their impact on schizophrenia and the studies done in this field. Genetic factors are having a role in the etiology of schizophrenia. There are certain studies where three basic methods are used: cytogenetic ones, community-based ones. So as a result of these three different methods, it was seen that genetic factors do have an important role in the schizophrenia's etiology.
Moleküler, buradaki slide İngilizce ama. Molecular genetic.
And the aim of the molecular genetic studies is to identify genes that affect inheritance. I mean, which gene causes schizophrenia and why they function in an abnormal manner. So maybe there are certain relations, and this was also found, I mean, the relationship between the genes and the disease. But the hereditary characteristics of the schizophrenia is not in coherence with the Mendelian pattern, the traditional pattern. It could be a polygenic hereditary character. So with regards to this disease, a major locus was tried to be identified, but we cannot specify this to date. Environmental factors do have also a role in the etiology. But what is the difference between genetics and epigenetics? Epigenetic, especially in molecular genetic studies, is just a different perspective, because environmental factors in schizophrenia are having an important role. We know that, and when the environmental factors go up to a threshold, and there are certain hypotheses stating that it might also result in schizophrenia. So in a way, how environmental factors can change the genetic structure, the answer can be given via epigenetics. In the DNA base sequence, there is no difference, but the gene expression changes. So we know that a mutation, deletion, or other abnormalities do not exist, but the gene expression changes. So these are called epigenetic mechanisms. Here,
sonuç gen ekspresyonu çevre.
So gene expression, thanks to environmental factors, do change, and this is not something permanent. It's not like a mutation or a deletion. These are temporary abnormalities, and they can be reversed. All endogenous and exogenous signals do have an important role in the integration of these signals, and genomic imprinting and cell differentiation and X chromosome inactivation are very important, and epigenetics also has a role in this, and dysregulation in the DNA methylation is observed. Environmental factors and epigenetics. We know that all environmental factors do have an important role in terms of schizophrenia: diet, medications, hormones, maternal care, and toxins, drug abuse. So all environmental factors that we know have an impact on epigenetic mechanisms. What are these mechanisms? One, histone modification. The other is DNA methylation. Histone modification is as follows. You know that these histones are proteins covering DNA, and then nucleosomes and chromosome structure is formed. And these histones, these proteins, are are more resin-analgenin, and they are prone to a chemical activation, acetylation, methylation, phosphorylation, and many chemical changes may trigger genetic changes. So the acetyl or methyls bound on histone protein do change their structure. Acetylation and methylation is the most common one in terms of histone modification. And then we have DNA methylation. Methyl is added to guanine, the Stosin Islands. So what about histone acetylation? Histone acetylation is a dynamic process, and it is catalyzed by histone acetyltransferase, histone deacetylases. So when there is acetylized histone, there are spaces and the gene is activated, but there's deacetylization, then transcriptor factors are banned and the gene activation is suppressed. So when histone is acetylated, then gene activity increases. In other words, here you can see histone acetyltransferase added acetyls, and on the left hand side there is the active gene, and the second is the inactive one. It is condensed chromatin, it's inactive. Histone methylation, which is a little bit more complicated. In histone methylation, it is based on which histone is methylized, and the outcome may change. For example, the third histone and in the methylation in the fourth group may increase the gene activity. However, 10, 9, 27 acetylation or says methylation does suppress activity. So it is also very important, the location of methylation. What about histone modification and its relation with schizophrenia? Were there any studies pertaining to that area? Yes, there were. Third histone, that is H3, and 17, protein 17 is high. This is quite in relation with the downregulation of metabolic gene expression, and the histone modification may be associated with schizophrenia. And the other was DNA methylation. DNA methylation is characterized by the methyl group transfer in CpG dinucleotides. So when a methyl is bound to DNA, then the transcriptor factors coming to DNA is blocked, so there is gene activity which is suppressed. So when there is methylized DNA, there are proteins bound on them, for example, LBP, for example, and MBP. So gene is silenced here in this respect when they are bound on those molecules. So DNA methyltransferases do indicate in this reaction, and methylating the cytosine, here you can see such an activity is suppressed. But this picture can be reversed. So this is the demethylized DNA methylase transferases. When they are deleted, here you can see that the gene is activated again. Global methylation and the level of global methylation is significantly low in schizophrenic patients, the study suggests. It goes back to 1960s and 70s. There were two studies that suggested the role of methylation. Methionine use actually worsened the symptoms in this group of patients. Then in 2002, Costa and his friends interpreted this study in 1970 as follows. The hypothesis of methionine transformed to SAM in the body and provide methyl donors dopamine, and therefore may trigger the manifestation of schizophrenic symptoms, because some genes, GAD67 and GABAergic genes, and they are hypermethylated, and the genes are silenced and they deteriorate these functions. This is an hypothesis. GAD67 or GAD67, and there are two genes, candidate genes, and that is RAIL genes or GAD67, and their methylation status was also observed, and it was found that in the promoter area, DNA methylation and mRNA level is low. But as over-acetylizing the histones, it might be a counter effect or an adverse effect. So in epigenetics, there are many mechanisms, and we have to have a holistic eye to all these mechanisms. If you just observe only methylation, it doesn't give you information about the gene, because there might be contrary or adverse impacts. So you have to be in the know of both the histone situation or status and the methylation status. There was this animal study that shows that with methionine, RAL and GAD67 expression was decreased. Again, these methyl binding proteins were observed in the same study. So RAL and GAD67 promoters, in these promoters, the methyl binding proteins binding also increased. GABAergic dysfunction may be due to this phenomena in schizophrenia. Here you see that MCP2 binding to methylated areas, and it silences the genes, and when you delete these, this gene is activated. RAL gene is also observed, and this is also in association with the GABAergic activities, and it was seen that there is hypermethylation and the activity is decreased, and there is no DNA methylation observed in the RAL promoting region. And in another study, it was suggested that up until puberty, there is no methylation in RAL gene, or it's very low, but after puberty, methylation level increases. So one gene has more than one promoting region. So for the gene expression, other transcriptor factors should bind to them, but one gene has more than one promoting or promoter region. COMT, again a candidate gene, is observed and is responsible for the catecholamine structure. MG COMT can have a hypomethylation, a hypomethylation, and there is MB COMT transcription increase in schizophrenic patients. Another study shows that COMT gene is also hypermethylated. DRD2 and DOT genes were also observed. There is no difference in terms of methylation. Serotonergic genes were also observed, and it was found to be hypermethylic. And BDNF gene methylation was found to be low. We didn't expect that. BDNF2 promoting region was observed in our clinic, and there were no difference between the patient and the control group. As I said before, there are many factors in epigenetics that we have to study, and it might be due to the fact that we didn't comprehensively observe that. The impacts of drugs on epigenetics, I mean those drugs that we use, drug abuse during childhood or trauma, infections, everything, I mean hormone might have an impact on epigenetics. All the environmental factors maybe are already resulting in this disease. This is the epigenetic hypothesis. And of course the role of drugs, for example antidepressants, thanks to the increase in methylation, might have an impact on that. Especially valproate, when combined in schizophrenia, inhibits, or in other words causes histone to acetylize and normalizes REN levels and GAD67 levels. And valproate also increases histone acetylation and DNA demethylation. So DNA is free from methylation, so it increases the gene expression. Maybe this is the idea of valproate. This is the action of mechanism of valproate. DNMT inhibitors, chemotherapy agents, and procainamide were used. Again, and they have impacts on GAD67. Nicotine increases in the NMT expression. Maybe this is an environmental factor, but somehow it might be also observed like a bad drug. It might have a different mechanism, because we have to also look into that. So in conclusion, epigenetic mechanisms play an important role in the central nervous system, dynamic interaction with the environment, and they have to be regulated. If it's too much or too low, it might also result in a disease. It's not possible to, of course, improve the genetic factors today, but we might only prevent its transfer to the new generation. But over epigenetic mechanisms, genetic factors can be controlled, and if we can do that, we can intervene to these patterns, and this picture can be irreversible. We can do that. Thank you.